right, now we have it.
Okay, happy to be here. Sorry we were slow getting going. I'm going to talk to you about my group's efforts to design and build a synthetic minimal bacterial cell. But before we get into that, I'd like to show you some family pictures. And I say family pictures because I think of the minimal bacterial cell we built very much as family. Here's just a bunch of my friends, and they are all clumped together in this scanning electron micrograph. And then we've got some ultra high resolution microscopy where we can see these cells in the process of division. And this is light microscopy. And then here's some negative stain electron micrographs. Again, the minimal bacterial cell. Here's a colony that these cells make on agar. And it looks a little bit like a fried egg. You see the yolk and then the edges of the colony around it. And then this is some cryotomography, and the yellow dots are the ribosomes. This is how we showed where the ribosomes were and counted them in the cell. And then this is some whole cell computational modeling of our minimal cell. And I know that this group is very much interested in the idea of computational modeling and applying mathematics to think about biology. This, to me, is perhaps the greatest moment of my scientific career, coming in and finding something so astonishingly different than what I expected to see when we videoed the growth of our minimal cell in microfluidic chambers. I'll talk about that in a moment. And then just this beautiful picture of a cell that I'd probably have a tattoo of, but I don't like tattoos. So if I did, it would be actual size. And this cell, so in 2010, my team, in our efforts to build a minimal bacterial cell, and I say my team, the group of scientists that I work with, there's no real ownership. We all work for Craig Venter. We built JCVI Syn 1.0, which was the first cell that had a synthetic genome. And then in 2016, we were able to finally minimize that genome and again publish about JCVI Syn 3.0. Now, I know you are Australian and New Zealand possibly, but if you were baseball fans or knew Kevin Costner's movie, people come out of the cornfield if you build it. When we built the minimal cell, right away, I started getting calls and emails, people who wanted to work with us to use this cell to investigate the first principles of cellular life. And now we have this ad hoc consortium of, the slide's a little dated, we're up to almost 70 now, of labs all over the world that are using our cell to investigate basic biology. So why are we doing this? The thing that has inspired me and my team, and in fact, I suspect many people in the world, is this fundamental question of what is life? And what we sought to do, because the Venter Institute is fundamentally a genomics institute, we wanted to use the lens of genomics to understand what genes are necessary for life. Or maybe another way of putting this is, what does a living cell, what tasks does a cell have to perform? Now, a lot of people have been trying to do this for decades with E. coli, but E. coli has 4,000 genes and is incredibly complicated by my way of thinking. I realize I'm a human with many more genes, but what we wanted to do is have the simplest possible system. The physicists and chemists in the 19th and 20th century, and even today, are making extraordinary discoveries about the nature of matter using the hydrogen atom. And their rationale is that what is true for hydrogen is probably also true for helium and uranium and lead, etc. So we believe that the minimal bacterial cell, JCVI Syn 3.0 or Syn 3.0, as I'll explain in a moment, can be the hydrogen atom of biology. So let me explain what a minimal cell is, because scientists have been wanting to make this since the 1930s, this idea of the simplest possible system that you can understand and get rid of as much of the complexity of the system as possible. So a minimal cell would be a cell that contains only the essential genes for independent growth under ideal lab conditions. No single gene can be removed without a loss of viability, and it has all the machinery for cellular life, so that ideally, if I knew the function of every gene, of every protein in the cell, I would have a real, perhaps near complete understanding of what it takes to be alive. And from this, we can go to computational modeling of minimal cell behavior, because much as I would love to have in my head everything that's involved in keeping a cell alive, it's too complicated. And so we really will need computational models to understand it, to predict the effects of eventually of environmental variations or how you might change the cell. Now, I live in the real world, and there are cells that take years to divide. We know there are cells that live in rocks that take perhaps decades to divide. So we want a cell that can divide every 2 hours without slowing the growth rate of the cell. And so where we went with this, we went to these organisms called mycoplasmas. Mycoplasmas are atypical bacteria in that, because of their obligate intracellular parasitic, their obligate parasitic existence, they can only live in one niche, if you will. They have been able to throw away most of the genes that are present in more conventional bacteria, like E. coli or Bacillus subtilis or Staphylococcus aureus or something. So Mycoplasma mycoides is a small bacterium. It's not the smallest bacterium in nature, but it's small. It is an opportunistic pathogen of goats. So you can see its size relative to E. coli or T. phage. It has a near minimal genome and minimal metabolic complexity. It's a total heterotroph. It makes nothing but more cells. No amino acid, nucleotide, or fatty acid biosynthesis. There's no cell wall, and it grows in really rich media. And in a couple of days, it produces these beautiful fried egg colonies. This one is blue because we put a gene called beta galactosidase in it, and it turns a chemical called XGAL blue. And this is a 1 millimeter plate you can see on agar. It divides in about an hour. And so to go from Mycoplasma mycoides to a minimal cell, we didn't want to just whittle it down. We wanted to build a bacterial cell from synthetic or a genome from synthetic oligonucleotides. And so we had to invent 3 technologies. We had to be able to go from synthetic oligos to genome synthesis. Now, when we started, the largest genome that had ever been made synthetically was about 8,000 base pairs. And so we initially made a 580,000 base pair genome of another species of Mycoplasma, and then later on made a 1 million base pair JCVI Mycoplasma mycoides genome. And we do this by taking 60mers and building cassettes that are larger, and then assembling those into a complete genome. And we would put a small piece of DNA that is a vector sequence for yeast, because what we had to do is build this and then move it into a yeast cell. Actually, we did the final assembly in a yeast cell, because we need to make a huge amount of this genome in order to do the last step, which is called genome transplantation, where we isolate this genome and install it in a suitable recipient cell to produce a new cell that has a synthetic genome. And once we built a cell, our goal was to see, we built this minimal cell, our goal was to see if we can understand using our life to produce a life synthetic software that would tell us what genes it takes to be able to complete all of the tasks. So you'll notice all of these tasks here. So for instance, all life on Earth needs lysine. Now you can synthesize lysine with about 30, 35 genes, and E. coli does this. But mycoplasmas don't synthesize lysine. Instead, they use one gene, a transporter, to import lysine from their environment, but they've still got to have lysine. So we believe that cells have to do all of these things in this list, and we hope we may learn that there's some other things that all cells do that we're at this point unaware of. We also used this process that is an engineering approach that synthetic biologists use, and sometimes it's called the design, build, test, learn cycle. But the idea is that we would, that we would design a cell, then from synthetic oligonucleotides, build its genome, boot up its genome using genome transplantation to make a new cell, and then based on the properties of this cell, if it worked or it didn't, redesign and do as many cycles as you need. Now, the heroes of design, and I think that this kind of silly because I have these two guys which work for me. Clyde Hutchison and Hamilton Smith are famous scientists. Ham Smith won a Nobel Prize. Clyde Hutchison twice was the last guy of the three guys who got a Nobel Prize to come off the list. So these two guys who are postdocs in my lab, which I think is kind of silly. Actually, they both, so Ham is retired, Clyde still works, and Chuck Merriman, who was a faculty person at the JCVI, they were the main people who did the design of the minimal cell. And the way we wanted to do this, there are two ways you can think about doing this. So we wanted to build a cell based on Mycoplasma mycoides to get rid of all of the non-essential genes. And one way to do this would have been to just whittle the genome away. That would have taken a long, long time. What we wanted to do was to design it based on what we know about the biology of Mycoplasma mycoides, then synthesize the genome and boot it up. So how do you figure out what genes in a cell are essential and what genes aren't? Well, first, basic biological knowledge tells you a lot. We know that cells have to have ribosomes. They have to have ribosomal RNA. But what Clyde invented in 1999, it was a program, a procedure, Clyde Hutchison invented this procedure called global transposon bombardment. So transposons are jumping genes. And here you have a transposon that expresses an antibiotic resistance marker. And so and by jumping, they will integrate randomly into a genome. If the transposon jumps into an essential gene, that cell dies. If it jumps into a non-essential gene, it will yield a living cell that is resistant to tetracycline. And then we can sequence out from the transposon in both directions to localize the site of the transposon and say that that gene is not essential. So the way we do this is we build this PCR product, polymerase chain reaction. It's a way of producing synthetic DNA from a template that has, it's about 800 base pairs, but this is its sequence. We put this DNA into, we mix this with a commercially available transposase, the enzyme that mediates the insertion of this into a bacterial genome. Then we introduce this transpososome complex into a culture of Mycoplasma mycoides, Syn3.0, the genome that is almost identical to wild type that we first synthesized by polyethylene glycol transformation. And we collect colonies. Now, the first set of colonies is P0, passage 0. There are about 80,000 colonies. And then this, you will find transposons in almost everything because cells will live for a while even after transposons go into essential genes because they're still residual protein. So P0 we don't use, but we passage all of these colonies into new growth media as passage 1, and then we grow that and then work our way up to passage 4. And we prepare DNA libraries from the cells in passage 1 and passage 4. Note that only fast growers are present in P4. So then we do a process where we cut the genomic DNA and then sequence out from the transposon to identify the sites the DNA inserted. And so wherever the DNA inserts, wherever the transposon inserts in this map, it's either a white box or a red box. And the white box is library 1 for passage 1. The red box is for passage 4. Now here you see, bear with me, an essential gene, CTP synthase. It has no transposons in it, no boxes, white or red. And here you see a gene that so if you knock it out, the cell will grow more slowly, meaning it needed this protein to grow at the cell's normal doubling time of every 60 minutes, because all you see are white boxes here. But a truly non-essential gene will have both red boxes and white boxes. And so here's a gene that we know you can knock it out without any loss in growth rate for the cell. And so what we produce is this, what we found is about half the genome is either essential or quasi-essential, meaning that that gene is necessary for rapid growth, and half the genome is not essential. And what we then did is built a genome based on the genes that were essential or quasi-essential. It's an interesting story. So we had been thinking about this, and at a lab meeting one day, Craig Venter comes in and he says, I want to make the Hail Mary genome. Now, a Hail Mary pass in American football is a desperate attempt to win the game at the end when you're behind, and you fling caution to the wind. You do it in the last seconds of the game, and you hope for luck. Now, a Hail Mary genome, we're going to fling caution to the wind because we moved our DNA synthesis to a company that Craig started, and they had some extra synthetic capacity. And so Craig said, do you guys have a minimal genome design? And I said, yeah, sort of. He said, well, give it to me because we're going to synthesize the genome next week. And so we said, I'll get it to you tomorrow. And we put the thing together and we hoped for luck. And so here it is. You can see that if this is the wild type genome, the Hail Mary genome is about half the size of the wild type genome, and it makes 432 proteins as opposed to 923, and it drops off a couple of RNAs. And the way we built this is in 8 segments. And we first built it as 1/8 minimized and 7/8 full size. And we booted those up, we transplanted them, and to our enormous delight, one of them worked, only one. But that told us that this approach would work. Now, interestingly, when it was time to write the paper about this, because all of our notes and everything talk about the Hail Mary genome, and we said, Craig, we can't say Hail Mary genome in a journal. But one of my guys said, don't worry, it's the hypothetical minimal genome. So that's how we got around that small but perplexing problem of not wanting to offend anyone. So the hypothetical minimal genome, we decided that we needed to have better transposon data. So we went back in and got a lot more transposon data, and we made a new design, which we called Reduced Genome Design 1. And if the wild type is a little over 1 million, Reduced Genome Design is about a little more than half that. And we built this, again, in 8 overlapping pieces, and we made, it's a 50% reduction. And so now it's time to build this thing. And the heroes here are Dan Gibson, who invented Gibson assembly, Ray Schwang, and Vladimir Noskov. And they first built the small synthetic pieces that were, so for our final genome, they were provided as, so initially it's in vitro, where we do oligo design and synthesis to make 1 kb pieces, and then these are assembled in vitro to make larger, we do error correction and make larger pieces to amplify these. We then move these into E. coli to make pieces about 7,000 base pairs. And then finally, we do the last step where we take the 7 kb pieces and assemble them in yeast to first make eighth genome molecules and then a complete genome. And the next step, so we built this again as pieces that are 7/8 wild type, 1/8 minimized. And we also made some combinatorial assemblies where we would mix in a tube all of the minimized pieces, or in a yeast cell, all of the minimized pieces and all of the wild type pieces, and yeast puts them together to make all different combinations of the complete circle. And so the next step is to test this thing that we built in yeast in genome transplantation. Now, genome transplantation, the idea is we will take a synthetic genome that has a small antibiotic resistance marker in it, and we will install this in a bacterial cell called Mycoplasma capricolum, which is a different species than Mycoplasma mycoides, but very, very similar. And so we treat the recipient cell with polyethylene glycol and calcium ions. And it goes in sort of like a chemical transformation, if any of you biologists remember this. Next, we return the cells to growth media, and the cells divide. They are transiently diploid. They will then divide, and we add the antibiotic tetracycline, which will kill any cells that have the native Mycoplasma capricolum genome, leaving only the ones with the synthetic genome that survive. And this is what we hope to work with. Now, transplantation, this is a sort of an aside point. It only works for non-Mycoplasma bacteria. And the world is to the state now that there are companies that will synthesize a complete 2 million base pair or more bacterial genome. But if it's not a mycoplasma, there's no way to boot it up and put it into a living cell. And so my team is working at trying to do this to extend the technology of genome transplantation. So right now, it's a huge bottleneck for the synthetic genomics field. Take E. coli with synthetic genomes. Jason Chen at Oxford and George Church at Harvard have built cells with synthetic genomes. But in order to do this, they have to swap the synthetic piece with its corresponding natural piece 50,000 base pairs at a time. And this is a process that can take months to years. So we can't be so lucky as that we chose the only bacterium in nature this works with, because genome transplantation for E. coli would work in a day. We understand the mechanism of genome transplantation, and we know the secret is in preparing the competent recipient cells. And we're going to do this. And I think this will be a huge boon to the field of building synthetic cells, because it will make it so much more practical for people to do this rapidly and quickly using the commodity now of huge amounts of synthetic DNA. So, and we're also going to do this in a machine rather than in having a human do it, because humans it is really touchy. Okay, so we built our minimal cell that was 7/8 wild type and 1/8 minimal. And to our enormous delight, they all worked. So here you see cells growing that are 1/8 minimal with each of the minimized segments labeled. And this was fantastic. All the cells doubled in less than 2 hours. And this was just super. So, but we didn't get a complete reduced genome. what we found is we got, again, all the 1/8 minimized genomes were functional and grew well. And we also saw that some combinations of the 8 reduced genome design segments grew well. So here one that had 5 of the 8 segments reduced made pretty good sized colonies, smaller than wild type, but it grew. And then another one you see even made tinier colonies. And we realized the problem was something called non-obvious synthetic lethals. So a pair of synthetic lethal genes means that both of them encode the same function, that this function is essential for cellular life, but neither gene on its own is essential. So you can delete either of a synthetic lethal pair. Now, think of it as you're flying in a 737. The right engine goes out, and the pilot will tell you, we're fine, we're going to land soon, but it's perfect. And then, or on another flight, you learn that the left engine goes out. No one on the plane they might be not too worried if only one engine goes out, but we all know that we're in real trouble if both engines go out. And that's what a synthetic lethal is. So for instance, we would know that the ribosomal RNA operon, which the Mycoplasma mycoides has 2 of those. So the transposons are going to hit both of those. And we know they're not both non-essential, so you have to keep at least one. But where it gets hard, if you have 2 proteins that look different, but they both have the same function that you're oblivious to, you have no way to know that those are synthetic lethals. So it was time to go back to the drawing board. And we took a genome that was half minimized and half wild type and did a transposon bombardment again. And based on that, we took 20, because what we expect is that genes that were previously not essential will now become quasi-essential or essential. And we found 26 of those genes in segments 1, 3, 4, and 5, which were in the non-reduced segments here. And so we took those, we did not take those out of 1, 3, 4, and 5, and we built a new reduced genome design 2.0. And we booted that up and we got a cell which we call JCVI 2.0. And it grew, it made a nice colony. It grew about half the speed of the wild type cell. And it made colonies about the same size. Kim Wise was the person who does all of this work. He's a famous mycoplasmologist. So we thought we were done, but Craig said he wanted us to go back to the drawing board for cycle 3. We did another round of transposon bombardment and got rid of 37 non-essential genes to produce RGD 3.0, which was built, it was viable. It has the smallest genome of any bacteria in the world that can be grown in culture. And so here's what it looks like, sort of, but this is what the cell looks like, this electron micrograph. And from the rest of the talk, what I want to do is talk to you about how we have gone from building a synthetic cell and developing the technologies to do this, to really looking at it and trying to figure out how this is going to teach us about life. So here's the cell again, but this is how I want you to think of it. Art by Swedish artist Urs Wellers. This is what we want to do, is take the salad apart and see what's in it. And so when we look at the genes in the minimal cell 3.0, about half of them are involved in the central dogma of biology: expression of genome information to make proteins and messenger RNA, and preservation of genome information to maintain and replicate DNA. Now, we also saw, unexpectedly, because people have been doing biology a long time, that there are still a huge number of genes that in this minimal cell, the kernel of biology that we have no idea what they do. We were not expecting this. This is a complicated figure, but bear down with me on this. So what we did, we took all of each of the 470 some protein coding genes in the minimal cell, and we did a blast search against the genome of these 13 different organisms. A blast search looks for sequence homology. If we saw, and so let's look at E. coli here. If we found that E. coli had, for whatever gene this is here, had a very similar gene, that gene would be colored blue. Whereas if a particular minimal cell gene didn't have a similar gene in E. coli, it would be a white spot. And so we did this for all of these organisms, from archaea to yeast, fruit flies, humans, plants, a whole variety of bacteria. And what we saw is that for the genes that we were highly confident of the function, they are conserved, they are highly conserved. And this blast was very stringent. If we made it just a little bit more relaxed, this would all almost be colored. So first we did it for the genes we knew what they were. Then we did it for the genes that we had a pretty good idea what they were. And again, you see that there is high conservation in bacteria, in all different kinds of bacteria, and then in plants, yeast, humans, Drosophila, a little less so in archaea. And then finally, we did it for genes where we have either a vague idea what they do, like they might be a transporter or a membrane protein, or no idea what they do. And what we found is that there were a huge number of genes that we don't know what they do. And this is what we've spent a lot of time working on, is figuring out what these genes, this third of the whole genome, what the functions of these protein coding genes are. And we've been working at this for about 6 years, and we've now reduced it from 149 to 78, and we're doing better and better. And AlphaFold, for instance, where we can predict protein structure based on its primary sequence, has helped a lot here. Now, one of the things that we have to remember is that mycoplasmas are evolving more rapidly than any other bacteria, meaning that, and when I say this, so this is a dendrogram of all bacteria, and what you are seeing is the length of this radius is proportional to how fast this organism is evolving relative to all these other organisms. And so here the mycoplasmas relative to most other bacteria, and you can see that they are the only ones that are really close to the edge here. And mycoplasmas, again, they are not primordial bacteria. They were like other bacteria, but they lived in such narrow, unchanging environments that they could throw away lots of genes, and including those genes were genes involved in repair of mutations. And so mycoplasmas are exploring sequence space. So there could be, among all these genes of unknown function, there could be some that are doing the exact same thing in mycoplasmas that they are doing in other bacteria. They just don't look like the genes in other bacteria. Or alternatively, there could be genes here, essential critical genes that are present from everything from mycoplasma to man, that are doing something that we are an essential thing that cells have to do, that we are completely oblivious to. And I think that's really kind of cool. And probably there isn't, but that we really might find that. So this is one of the real questions that we're working on. Genes that may perform the same function may no longer look similar, and there could be genes that are critical to all life on Earth that are doing something so, incredibly vital, and this function we are just oblivious to. I'm going to go through a few vignettes. So, minimal cell morphology and the evolution of cell division. Li Ji and Kim work at JCVI. James Pelletier at the time was an MIT graduate student. Now he's a postdoc in Madrid. And Elizabeth Strahowski they're both physicists, but Elizabeth is a physicist at the US National Institute of Standards and Technology. And what prompted this is we realized that some genomically reduced strains exhibit amazing variation in cell size and shape compared to wild-type Mycoplasma mycoides. So here's wild-type Mycoplasma mycoides, cell diameter about 400 nanometers. And this is a minimized strain where it's 7/8 wild-type, but segment 6 is minimized. And these two micrographs are at the same scale. What's going on here? And what we decided to do is to try to watch the cells grow in microfluidic chambers. And so we build these devices that have tiny little chambers. You see the dimensions that are linked to a chamber and this channel. And we flow cells through the channel and a small number that are in growth medium. What we hope to do is get one cell per chamber. And we're pushing fluid through here, comes through the channel and out. And the idea then is if you can get one cell in the chamber, we can do video micrographs to see how the cells grow and what's really going on. So here's a chamber where we're seeing JCVI Syn3.0 over about 20, 30 seconds. So first we looked at wild type, Syn 1.0. And this is bright field, and this is cytoplasm, where we have the cytoplasm of the cells. We're producing a red fluorescent protein in it. And so you can see the time, and this is sort of what we expect. Here's a video. Again, M. cherry, the fluorescent things, but these are 400 nanometer spheres, and they gradually grow and fill up the chamber over about 13 hours. This is what we expected. This is it's what we expected. And you can look at bright field or the cytoplasm that is with M. cherry. We can stain the DNA with this blue dye, or we can stain the membrane with this green dye. This is what it looks like. Okay, get ready. This was the great shocker of my scientific life. We come in and we look at the video. This is a minimal cell, 3.0, and this is over 8 hours. So we start with one cell, and we see it grow to make this piece of spaghetti. And then there are these holes in here. It's like, what is going on? And in science, you live for this. You come in and find something so different than what you expect. It's a little shocking, but this is something that will hopefully change the way that you think of nature. So they have unexpected morphologies, bright field. Here we see this hole that is filled with DNA, with membrane. This is where we were. And what we realized that this is not what we expected. Now, we realized also that we had a minimal mutant that was built in the process of constructing the JCVI Syn3.0 that had an additional 19 genes. And in the microscope, this made normal-looking cells. And so we thought we would look at those 19 genes to see if it might teach us why the cells grew so aberrantly. And at first we went, aha, here are two known cell division proteins that are involved in making a lipid protein septum between growing cells as the two daughter cells divide. And we said, I bet if we add those back in, it will make to JCVI Syn3.0, which divides so aberrantly, it will look normal. It didn't. We added those back, and it didn't restore the cells to normal size. So now we come up with about a year, 18 months of combinatorially testing things. And what we found is it was no one protein, it was 7 proteins that were required to restore the cell to normal cell growth phenotype. and 5 of them, we didn't know what they did. And so we are working now to try to understand this process. But we also realize that we may have accidentally created cells that recapitulate aspects of what primitive cells may have looked like. And this is when we got an unexpected note from a couple of paleobiologists from Poland. They wrote my collaborator, Elizabeth Strachowski, congratulations on your article. We were surprised to find that your minimal cell is similar, if not identical, to the pattern of cell divisions we see in microbiota from extremely old Paleoarchean chert, which is a sedimentary rock in South America, 3.4 billion years old. And here's our cells, and here's their cells. And I find this a little bit of a struggle, but they've spent years looking at this stuff, and they liked it enough to write us. Now, we realized that we hadn't intended to make a primordial cell, but what we may have done is make something that is the way life on Earth was before invention of all the cells that we know of on Earth practically. So all cells on Earth have FTSZ with a tiny number of exceptions. What if before modern cell division evolved to produce a protein and lipid septum, instead you got something like this, where you got a vesicle encased in lipid, and as the membrane kept accumulating more membrane chemicals, phospholipids, the cells eventually divide without making a septum, but based on the physics of membrane curvature. We suppose that this might be what's happening, and we're doing some experiments now to think about how transporters may be involved and the forces of membrane curvature. And so we hope to get back with you before too long to maybe let you see, to get a better idea what modern cells, what ancient cells looked like before we developed modern cell division. There are homologous proteins to FTSZ and CEPF in our cells, in all but a tiny number of bacteria that are unusual and probably threw away CEPF and FTSZ. Now, you guys, I know many of you are thinking about computational modeling. Shortly after we published The Minimal Cell, Zan Luthi-Schulten and her team at the University of Illinois called us to see if we could work with them. They had been thinking about modeling E. coli, but they realized that the minimal cell was a much better cell to cast into a complete computational model. And so we have been doing this with them for the last 6 years. A few years ago, we published a model made by now scientists at Maastricht University in the Netherlands, Marion Brewer and Zan, this whole cell flux balance analysis computational model that takes into account everything that at the time we knew about the minimal cell, 155 genes. And for instance, the ribosome involves 3 ribosomal RNAs, and about 60 ribosomal proteins. That just counts as one entity in this model, but we have 336 reactions, all the metabolites that we know of, and the model reproduces the experimental doubling time of the minimal cell. The modelers have also gone so far as to show where they see the ribosomes are, where they see proteins are, and to make this active computational model that tries to take into account eventually every atom in the cell. But looking at this aspect of metabolism, the model is very helpful to suggest experiments. For instance, our cell lacks the E1 subunit of pyruvate dehydrogenase, but the model, because the gene is not essential, but the model predicted that if we added it back, the cell should grow 18% faster. And so my teammate at the time, Andras Cook, added the E1 genes back to the minimal cell, and the experiment didn't quite work out. So when Andras added it back, it only grew 16% faster instead of 18, and we chastised the computational biologist, but in reality, that's a pretty great result. Another thing that the computational model predicted is that although there's no gene in the genome that looks like carbohydrate metabolism protein transaldolase, for the model says that you have to have a transaldolase for the genome to divide in 2 hours. Without it, it would divide in 8 hours. And so Andras decided to figure out how we can do this. What we did, and this is what transaldolase does, it does this reaction. Andras added a Bacillus subtilis transaldolase to the JCVI Syn3.0 to the minimal cell. We put that back in, so now we assume that the cell has a Mycoplasma transaldolase, which is quasi-essential, and a Bacillus subtilis transaldolase, which would be non-essential. So now, since it's got two of these things, neither gene would be essential. Then we did a transposon bombardment and to see if we can identify the landing pad. And what we found, whoops, too fast. What we found is a gene that was previously quasi-essential and now is non-essential. And Bacillus subtilis transaldolase was also non-essential. But this gene that we identified looks nothing like a transaldolase, but this is one way that you can figure out the functions of unknown proteins. And we're going to test this protein to see if it really has transaldolase activity. Sorry, I'm going slow here. Let me speed up a little bit. We have another example of how the cell may recapitulate what life was like before the invention of chromatin. So bacterial protein HuPA is an essential protein involved in chromosome condensation. E. coli has 30,000 copies. Wild-type Mycoplasma mycoides has 6,000 copies, but the minimal cell has 28. Now, what you can see here, if this is the HuPA gene, this gene in Syn3.0, GPSA, is not essential. So we took that out to make Syn3.0. That gene's gone. And we think that in doing so, we messed up the expression of HuPA. And so in theory, if we put this back in, it might restore the normal level of HuPA synthesis, the histone-like protein. But we also noted that Remus Dame at Leiden University in the Netherlands did what's called a chromosome conformation capture study, where you take genomic DNA from a bacterium and you chemically cross-link it so that any pieces of DNA that are bound to each other will stay cross-linked. Then you sequence those pieces and you find that in Syn3.0, there was essentially none of it. But in E. coli, the reason we know that there's a lot of cross-linked DNA that is not immediately adjacent to this line of the main if this is genomic DNA, these are places where you have DNA from two regions of the genome being together. And so in the minimal cell, there's basically no long-range chromosomal interactions. And we think that the HuPA may be mediating that. And so we're going to put GPSA back into the genome and then do this chromosome conformation capture study and see what happens. So we're excited about that. We're trying to build living synthetic cells from non-living parts. So we're going to make the idea is we will take a cell-free extract from living Mycoplasma capricolum cells, where you blow the cells apart and collect the cytoplasm that has ribosomes and messenger RNA, and we get rid of the genomic DNA and the membrane. And so we'll put into this method, this extract that does that has RNA transcription and translation of proteins. And we'll put those in tiny little vesicles, lipid vesicles. And we'll also add a plasmid that has a small number of genes to make transporters and a few things. And so we'll put this in a solution where the cells will survive for about a week if you have nutrients and can have exchange of metabolites with the inside of the cell and the outside. And so the goal then is we will take those vesicles that have the transcription translation reaction, and we're going to try to put a genome into them and see if this boots up a living cell. So we're excited to see if this happened. And we spent 3 years trying to figure out how to make a cell-free extract, and absolutely nothing worked. This was supposed to be the first, like, 1 day of the project, but 3 years produced nothing. But finally, when we had even published a paper saying we couldn't do it, a graduate student working on the thing at Radboud University figured out, using an old method that molecular biologists had used, how to make this work. So we're really excited to get going on this again. And almost the last thing, we have this idea that the minimal cell might be able to be used as an anti-cancer system, because we know that the minimal cell, it appears to be invisible to the human immune system. And so we could put genes into it that might enable us, well, it has a bunch of features to make it invisible to the human immune system. But the idea is, and here you can see the wild type cell exposed to human phagocytes that are readily just gobbling up these little dots. But here, the minimal cell, the human phagocytes don't see them at all. So what we hope one day, and this is the statistics on this, but the idea behind this is that, imagine I've got a minimal cell and I've engineered it to express proteins on the cell that could kill a cancer cell. Say this protein called TNF-related apoptosis-inducing glycan, which people have shown can kill cancer cells. And we're going to put those on the surface of the cell. And then we'll also put an antibody that binds to, say, human colon cancer. And so the next thing is, if you have colon cancer, and we would treat a patient or a mouse with colon cancer with our cells by injecting them in. And so the immune system is not going to be activated, and the cells can easily be killed with antibiotics. They're exquisitely sensitive to antibiotics because of the minimization process. And so what we would hope is that you would get rid of the cancer and the patient would be cancer-free. The cells would readily die when you treat with antibiotics. And we're doing those experiments now. So this might be a really exciting use as an experimental chassis. Ours is the simplest organism and the most genetically malleable organism on Earth. It can grow in a defined media to facilitate metabolism studies. We know the functions of the vast majority of the genes, and more than a larger fraction than for any other cell on Earth. It has a small cell that is readily amenable to microscopy. We have a community of researchers working, and it is non-pathogenic. So with this, we hope to be able to do a lot of things, like we've got a tool to investigate the first principles of life. We have our idea to get a cell where we understand what everything does, and we know really how cells work. And we are developing algorithms for genome scale redesign, so that based on what we've learned from the minimal cell, we can build more complicated organisms to solve human problems in agriculture, industry, or medicine, or research. I'm late in my career. I'm not concerned about prizes or publications. We don't do tenure at the JCVI, but I seem to be able to keep my group funding. My ambition in life is to understand the difference between a bag of chemicals and a bag of proteins, a lipid bag of proteins, and one that's alive. Now, I understand about glycolysis, and I've got a pretty good idea how protein synthesis works, but to really understand exactly what's going on at the nanoscale, that's what I want to achieve with this work. And I know that folks in your group, the people listening, some of you guys are thinking about modeling or thinking about building cells. If you're interested in getting the cell to work with, write me. I'm happy to send it to you. If you want to think about doing computational modeling, I'd love to do that. Right now, I've got a large community of collaborators, people at my group, at MIT formerly. Pelletier has moved on. Johns Hopkins, they're now at Harvard, they moved. The company Synthetic Genomics Incorporated, which is now Telesis Bio, the U.S. National Institutes of Standards and Technology, a agricultural research institute in Brazil, Technical University of Dresden, lots of places. This is an extraordinary effort we're on. And with that, I'll stop and hopefully get some questions.
Thank you so much, John. So there's one question already in the chat. I'll read it out. Hi, for the minimal cell that was computationally modeled, what do you estimate is the influence on the cell of random kinetic fluctuations in reaction speed and or other interactions between biomolecules? So to what extent is it stochastic, I suppose?
I think it is certainly to some extent stochastic because protein synthesis, there's a certain, yes, we have transcriptional regulators and translational regulators that are at slightly different strengths for each gene or each operon, and it will produce cells that have pretty much the same mean composition of all of the proteins, and transporters will keep a mean composition of the metabolites about the same, but not exactly the same. And so at some point, I know that I'm going to start using our institute's single cell transcriptomics analysis systems to see about variations in messenger RNA from cell to cell. And hopefully someday we'll have single cell metabolomics. I think it's an interesting problem. I don't know anyone working on this at the time, at the present.
And for me, a question was, so with real naturally evolved cells, you have some observations which have been puzzling people for a long time, and we can't even decide if they're paradoxical or not. So for example, the faster bacteria like E. coli grow, the larger the cell size seems to be, on give or take. Is that the same for the minimal cell, or is that more, does it behave more rationally?
It's interesting. So recently a paper published in Nature where I was a co-author with Jay Lennon and his students from Indiana University. And we identified, so we took the minimal cell and the wild type cell, and we did adaptive laboratory evolution. And so when we passaged the cells every day, where you went through about 2,500 cell division cycles in the minimal cell, we found that the cell evolved, both cells evolved to grow about 20% faster. And one of the genes, and then we identified the mutations that accumulated. And one of the sets of genes that we noticed in both the wild type and the minimal was, one of the things we noticed in the minimal cell is that if you put these mutations, there were mutations in FTSZ. And so we synthesized an FTSZ gene with those mutations, put it back into the cell, and yes, it grew faster, but it made smaller cells.
Okay.
Slightly smaller cells. Whereas the wild type organism grew faster over time, and it made cells that were 80% larger in volume. Again, that's not going to be a big diameter difference, but still So one cell grew faster and got bigger, and the other cell grew faster and got a little bit smaller.
So the paradox, as I would see it, persists at the natural cell, the wild type, but not at the synthetic cell.
Yes, yes. But I'm sure there are other examples like this, and this is something that I would love for someone to start playing with.
Yes, no, I can see that. And I think I give people more chance to put something in question and answer. But one question I have, and that's largely because you're in Craig Venter's sort of institute, which doesn't really limit the resources that you have at your availability. What would be the difference between a minimal cell and a minimal eukaryotic cell, John? Has anybody thought about this, or is it?
First, I need to correct one thing. So we are, I mean, yes, it's Craig's Institute, but all of the faculty, we compete for grants like everybody else in the US.
Oh, I'm so sorry to hear that.
So
yeah.
if you don't get grants, your job goes away.
Okay.
Now, a few years ago, with funding from the US Department of Defense, their Defense Advanced Research Projects Administration, they funded us to try to minimize the fastest growing known eukaryote, which is a yeast called Cluveromyces marxianus. It grows basically twice as fast as yeast. And we worked on this. They gave us $900,000 in a year to work on this. And we got some progress, but not enough they lost interest in this. I know that the Yeast 2.0 consortium is now starting the Yeast 3.0 consortium, where they hope to minimize this eukaryote.
Yes, okay. So there have been no more questions. And you mentioned Hail Mary. We've got an apocalyptic weather situation brewing over Melbourne, so with rumbling of thunder in the background. So thank you so much, John. The video of the lecture will be on the Theory of Living Systems website. And I think we'll just email you with requests for models, and maybe resume the conversation, John, if that is okay with you.
That would be terrific. That would be terrific. I would enjoy that very much.
Thank you so much for your time, sharing your wisdom and your exciting work, and giving us this insight into what makes life tick at the minimal level. Is fascinating.
If anyone wants to play in our sandbox, send me an email.
I'm sure we will do this. Thank you so much.
Lucy and Michael, thank you very much for inviting me. I really enjoyed this.
Thank you.
Thank you so much.
Okay. So you're
Bye now.
Still, John. Okay, if you stop it will stop for everyone.
Okay, and end.
Okay, perfect.